Hello everyone, I am Dr. Payal Gulati working as MK Bhan Young Researcher in Gene Regulation Laboratory at NII, and my today's topic of presentation is Electrochemical Immunosensors Fabrication for Oral Cancer Detection in Patient Samples based on Screen Printed Electrode. And I have carried out this work under the supervision of Dr. Pratima Solanki in JNU. Now, what is oral squamous cell carcinoma? It is a carcinoma that begins at growth in mouth and then extends to other part of mouth involving lip, tongue, cheek and floor of the mouth, hard palate and then eventually leading to pharynx. This oral cancer is reported to be the second largest cancer in India, and the global incident report in India presents the 77,000 new cases of oral cancer and 52,000 death cases. This oral cancer is detected with the help of biomarkers. Biomarkers are the biological indication of the disease condition, and it is used to monitor or recognize the alteration in the cancer cells. The proposed biomarkers for this work are IL-8, CIFRA, and P53, which are used for the immunosensor fabrication. And the principle for immunosensor fabrication is immunosensing, where one of the species antigen or antibody is coupled to the signal transducer and which detects the binding of the complementary species. For the immunosensor fabrication, my first step includes the fabrication of conductive carbon-based screen printed electrodes. For screen printed electrode fabrication, first initial step is the synthesis of conductive carbon ink, which is made using gum arabic graphite powder in DI water, and thereafter this ink was printed on a substrate, on a hydrophobic substrate, using the screen printing stencil and Graphica screen printing machine. For this, the substrate optimization was also carried out, where three substrate was chosen. First is the PET substrate, second is the ivory substrate, and third one is the hydrophobic sheet. And all of these sheet contact angle measurement was carried out to recognize the hydrophobicity, and the hydrophobic sheet showed a theta value of 124 degree in comparison to PET showing 68 degree and ivory sheet showing 110 degree. And also after ink printing, the cyclic voltammogram of all the three electrodes were taken, which clearly depicts that the hydrophobic substrate shows the well-defined oxidation reduction peak in comparison to the PET substrate and the ivory sheet. This is a schematic showing for immunosensor fabrication of all the three proposed biomarkers. This is a three electrode system where center is a working electrode which is modified with the antibody and BSA and eventually with antigen for the sensing purpose. As I have already discussed, the three proposed biomarkers for oral cancer detection is CIFRA21, IL-8 and P53. All of them have their own clinical significance for the oral cancer detection, as CIFRA21 is used for the oropharyngeal cancer detection. IL-8 is used to monitor the transformation from precancerous condition to the malignancy, and P53 is showing the mutation present in the 17p chromosome in the DNA of the patient. Now, the fabricated scene printed electrodes were characterized using scanning electron microscopy, where the micrograph shows the conducting path of the graphite electrode without any break, and EDS spectra shows the presence of carbon and oxygen, which confirms that graphite was used for the screen printed electrode fabrication. Further, the studies were also confirmed with the FTIR spectra as shown in figure 5. The peaks representing COH and CO2. These represents the porosity and hygroscopic nature of the graphite. And further, the modified electrodes, that is screen printed electrodes which were modified for immunosensor fabrication, they were also characterized using FTIR. Now, figure 6A and B represents that the FTIR spectra in which the bonds obtained, peaks obtained, which are characteristics for the amide bond and CN bond and primary amines, which confirms that the antibodies and BSA are attached to the electrode surface. Further, for the experimentation purpose, for the immunosensor fabrication, antibody concentration optimization was also done, in which it is clearly shown that 10 microgram per ml shows the highest peak current. Therefore, it is considered to be the optimum value for immunosensor fabrication. Also, the pH optimization study was done, in which the pH 7.4 shows the highest peak current, which was considered for the optimum pH value for experimentation purpose, and also it is considered as a physiological pH. Now the fabricated immunosensor results are also shown here in this slide, where this slide shows the CIFRA biomarker results. Initially when we modify the SP electrode with the antibody and BSA, we could see in figure 8a, after antibody and BSA immobilization, current was enhanced, and then when the sensing was done with different CIFRA biomarker concentration ranging from zero point 0.1 nanogram to 20 nanogram, as the concentration was increasing, the current was also found to increase, as shown in figure 7a, and thereafter interference study was done, as shown in figure 7b, where CIFRA 21, after forming bond with the anti-CIFRA 21, resulting in immunocomplex formation on the electrode surface, resulting therefore decreasing the current, and all other was acting as interference, which did not show any specific binding with the antibody molecules. Thereafter, the electrode efficiency was also tested with the patient serum and saliva samples, as represented in figure 7c and d, and also the electrode repeatability and stability test was done, as shown in figure 8c and d, and electrode was found to be repeatable with successive 5 readings, and also stable for the 5 weeks. The control study was also performed, as shown in figure 8b, where no trend was found, as antibody was missing on the electrode surface. Similarly, all studies were done with the isolate also. Only difference was the sensing concentration, that is from 100 picogram to 2000 picogram, and rest all the results were obtained similarly in case of CIFRA biomarker. And thereafter also another biomarker was tested, that is P53. Here the only difference of the sensing range was 1 picogram to 5000 picogram, and rest results were obtained in a similar fashion as in case of isolate and CIFRA biomarker. Now this is showing the conclusion of my work. The fabricated immunosensors showed the respective LOD and sensitivity as shown in the table, and the conclusive remark of my work are: the conductive graphite-based carbon ink was made in laboratory, successful fabrication of screen printed electrodes, successful fabrication of immunosensor on screen printed electrodes, and also the efficiency of screen printed electrodes were tested via patient sample testing with both serum and saliva samples. And I would like to acknowledge few people for this work. Firstly, I'm grateful to DST for funding this project, and thereafter I'm grateful to my supervisor for her continuous support and guidance throughout this work, that is Dr. Pratima Solanki, and I would like to thank my colleague for their support and cooperation, and also I would like to extend my vote of thanks to Dr. Alok Thakkar and Dr. Kingston in AIMS for providing patient serum and saliva samples, and also I would like to thank Dr. Renu Sharma for providing ELISA results to verify the data. Thank you all.